subject of this segment is going to be enzymes. Enzymes are proteins that have a specific function, and what they do is they catalyze organic reactions to make them occur much quicker. And when we look at an enzyme will cause a reaction to occur approximately between 10 to the 6th and 10 to the 9th times quicker. That's between 1,000,000 and 1,000,000,000 times quicker, which means that something that would take a day to occur would occur in the blink of an eye. So when we look at this, it allows for rates of cellular reactions to speed up phenomenally, which allows for larger organisms to occur. It says enzymes have unique characteristics of shape and specificity and function. Most of the time we call these things lock and key type things, that they only work on one particular material, but you may have other materials that could be affected by it, or you may have more than one enzyme that would work on the same material. Enzymes tend to be much larger than their substrates. That's so that they can grab hold of the substrate and manipulate it and cause it to come apart. Interestingly, it says enzymes are not used up or permanently changed by the reaction, which means the same enzyme can do the same process over and over and over again unless it becomes damaged. They are affected by temperature and pH, high temperature, low temperature, alkaline pH. Just like everything else, there is a optimum temperature, there's an optimum pH that these things work at. You modify it much, it changes. They may be affected by osmotic potential, and it says they can be regulated, which means we do have ways that enzymes are instructed to slow down or speed up particular kinds of reactions. When we look at enzymes, it shows that we have an apoenzyme, and the apoenzyme is the proteinaceous part of the enzyme. It says there may be a metallic cofactor, which could be a copper ion, it could be a magnesium ion, it could be any sort of ion that is out there that we would consider to be a metal. And it can have a coenzyme, which is a non-proteinaceous part of it, but the coenzyme may have to bind on. In here it shows metallic cofactor, enzyme, and both of them on different apoenzymes. So when we look at these, enzymes may have different constituents to them, and if it needs a metallic cofactor and you don't have enough of it, the enzyme may not function properly. When we deal with enzymes, it shows you a substrate. The substrate comes in, the substrate gets attached by the enzyme, and as it does it, it causes the substrate to break apart into the two products. If a material does not fit in, the enzyme will not affect. So you've got something that has a similar type cap to it, but the stem on it's a little bit different. The enzyme's not going to function on that. You may need a different enzyme to do that particular reaction. So when we deal with these are very specific in what they do. This part's very interesting because what it talks about is it talks about two types of enzyme. One is a constitutive enzyme and one is a regulated enzyme. The constitutive enzyme is produced, you add more substrate to it, you get no change in the amount of enzyme or the reaction at which it works. It just works at a constant rate, and if you've got too much substrate, it's not going to do much. If you've got a regulated enzyme, it says if you add more substrate, the enzyme will actually be induced to produce more enzyme. Or if you remove substrate, the enzyme would be repressed, which means the genetic part of it would not produce more enzyme until you had more substrate. And we're going to look at this in part when we look at DNA, because with DNA we have repressors. Remember, enzymes come from DNA because the enzymes are proteins, and we have repressors and operators in there that turn on or turn off certain pathways depending on whether or not you've got substrate. Because there's no point in spending a lot of energy creating an enzyme if the enzyme is not going to be used. This shows you multi-enzyme systems. And what you're talking about here is basically these are going to be biochemical pathways. And we're going to look at some of these. It shows over here that we've got glycolysis, we've got Krebs cycle. It says we've got amino acid synthesis represented on here. Glycolysis is a linear pathway where A gets you B, gets you C, gets you D, gets you E. Every arrow represents a different enzyme. In a cyclic pathway, it says U gets you V, And again, there's an enzyme that does that. At times you have inputs, and every time you've got a change from one to another, you're going to be using a different enzyme. And then at certain places, you've got products that can be removed. You can have branch pathways. Divergent type, where it says M will get you N, but then N splits into O and P, and you have two parallel pathways down here. Or you can have a convergent pathway where you make one thing on one side, one thing on the other side, and then they come together and they make something like an amino acid. So when you deal with amino acids, you've got different parts that come together to make these happen. So enzyme systems become very important because these become our metabolic pathways. We're going to talk about glycolysis in the Krebs cycle when we get to the part on respiration. Enzymes also have inhibitors. And if you take a normal substrate, you put it on the enzyme, you get reaction product. If you have an inhibitor that gets picked up that's got a similar shape, all of a sudden the inhibitor is going to bind, and when it binds, it blocks the enzyme from doing anything, and it may incapacitate the enzyme entirely. You can have non-competitive inhibition, and this is where you have a product that will bind to an enzyme, and we call this feedback. And normally this occurs if you got too much product. I mean, think about the way we do things nowadays as far as shipping material. If you go to the supermarket, they've got to have the material right there at that time. If you buy it on Amazon, it gets shipped out to you overnight. You can have it the next day. When you look at it in the supermarket, what do? They scan everything that goes in. And if something is not going off of the shelves, they don't buy more of it until what they've got goes off of the shelves. That is feedback. In here it says you've got reaction occurring, you get the production of product, you're not using up the product then attaches to the enzyme and it blocks the action of the enzyme so that it doesn't work, and it will continue doing that until the product gets used up, at which time this regulatory molecule, which is your product, will come off and allow the pathway to regenerate itself, so to speak. So when we look at enzymes, enzymes have feedback in there as well. This part becomes interesting. We're going to go into this when we get into the DNA part. It says DNA is transcribed to RNA. Messenger RNA is translated into a protein. Protein folds to form functional enzyme structure. The enzyme structure plus the substrate gives you the product. But if you've got excess product, it says the excess product could bind to the DNA and shut down the system so that you don't get any further enzyme production as long as you've got excess product. When the product is used up, these products over here will release from the DNA, and all of a sudden you can start the whole process all over again. Again, we're going to look over that again when we get into DNA. So when we look at enzymes, enzymes are extremely important. They are proteinaceous in nature. They have optimums as far as temperature, as far as pH, as far as substrate concentration goes. They undergo feedback so they can be regulated not to produce too much of a particular material. And they are extremely important in all metabolic pathways that we've got.